You know, in labs all over the world, there's this one process that happens every single day. It's the absolute first step for pretty much everything we know about genetics, from solving crimes to curing diseases. So, how do scientists actually get the code of life, DNA, out of a tiny microscopic cell? Well, that's what we're unlocking today. I mean, really think about it for a second. DNA is the blueprint for all living things, right? And it's packed away inside these impossibly tiny cells. So, before you can do anything with it, you've got this huge puzzle. How do you get it out? How do you gently pull out that super delicate strand of code from all the other gunk inside a cell? That's the challenge we're tackling. And believe me, this one process matters a lot. It's basically the starting gun for so many world-changing fields. For criminalistics, it's the genetic fingerprint that cracks the case. In medicine, it's how we can diagnose diseases. In biotech, it's the key to inventing brand new therapies. And for a researcher, it is literally the gateway to understanding life itself. Okay, so what's really fascinating here is that for all its importance, the basic process can be boiled down to a simple three-step recipe. It's like a universal guide that scientists follow to get their hands on that precious blueprint of life. So let's walk through it. First things first, you have to break the cell open. Think of it like a microscopic heist. The official science term is lysis, which pretty much just means to break down. To get to the treasure inside, the DNA, you've got to get past the cell's security system, its outer membrane. So how does lysis actually work? Well, you start with your sample, could be blood, saliva, maybe some plant tissue. Then you add a special solution, which usually has a detergent in it. And this works just like dish soak cutting through grease on a pan. It just rips apart the fatty membranes of the cell and the nucleus. And bam! Everything that was inside is now released. The DNA is suddenly floating free in this big soup with all the other cell parts. All right, the DNA is out, but now we have a new problem. It's totally invisible and mixed in with everything else. This is where step 2 comes in, and it's pretty magical. First, scientists add salts, which helps those long DNA strands start to find each other and clump together. Then comes the really cool part. You pour in some cold alcohol. Turns out DNA hates dissolving in cold alcohol, so it suddenly crashes out of the solution and becomes visible. For the very first time, you can actually see it with your own eyes as this beautiful, kind of ghostly white, stringy substance. And finally, we just have to clean it up. That clumped up DNA is carefully pulled out and given a quick rinse with 70% ethanol. This just washes away any leftover salts or other junk that might be hanging on. And what you're left with is a clean little pellet of pure DNA, totally ready for whatever amazing science comes next. And that's really the recipe. You break it, you separate it, and you purify it. But, of course, biology is never quite that simple. While that three-step recipe is the basic idea, scientists have to tweak it all the time depending on what they're working with. Because, yeah, one recipe definitely doesn't fit all. So where does it start to get tricky? Well, for starters, what happens when the cell you're after has more than just a soft little membrane? What if you're up against a wall? And this shows you the challenge perfectly. An animal cell, like one from inside your cheek, that's easy mode. It just has a soft membrane that a simple detergent can pop right open. But a plant cell, oh, that's hard mode. It's protected by this really tough outer wall made of cellulose. Getting through that thing requires a much more aggressive approach. So, to deal with these different challenges, scientists have a whole toolkit. For something tough like a plant, they might go mechanical, and I mean literally grinding or crushing it up. For something softer like a blood cell, the chemical approach with detergents works like a charm. And for some super tough bacteria with reinforced walls, they bring out the big guns, special enzymes that act like little molecular scissors, just snipping that wall to pieces. Okay, so you've broken the cells, you've separated the DNA, and you've cleaned it up. You're done, right? Not so fast. Now comes what might be the most important step of all, quality control. Because just having a pellet of something isn't good enough. You have to know if it's pure, usable DNA. This is the question every single scientist has to ask themselves. Is this sample I'm holding pure DNA, or is it contaminated with leftover proteins and other stuff from the extraction? Because if it's not clean, those contaminants can completely ruin whatever you try to do with it next. The main tool for this job is called a spectrophotometer, and the idea behind it is actually pretty brilliant. It shines a very specific wavelength of light through the DNA sample to see how much gets absorbed. You see, DNA and its biggest potential contaminant, protein, they absorb light differently. So by comparing how much light gets absorbed at the wavelength DNA loves versus the wavelength protein loves, you get a ratio. And the magic number that scientists are always hoping to see is right around 1.8. If that number comes back, say way lower than 1.8, it's a huge red flag. It immediately tells the scientist, hey, your sample probably has way too much protein still mixed in, and they might have to go back and clean it up again. Another really powerful quality check is called gel electrophoresis. This technique lets you actually see the DNA. It confirms that, yes, DNA is present, but it also crucially checks its integrity, making sure the DNA strands are long and healthy and not all shredded into useless little pieces during the extraction. So let's bring this all together. We've gone from a single cell all the way to a pure sample of its genetic code. This journey, from the lab bench to real-life applications, is exactly why this seemingly simple recipe is so incredibly important. At its heart, it always comes back to these three basic steps. You break the cell open, you separate the DNA out of that cellular soup using salt and alcohol, and you purify it, washing away anything left over to get a clean, workable sample. Break, separate, purify. And this one fundamental process, repeated in labs all over the globe, is what makes everything else possible. It is the absolute, non-negotiable first step on the path to sequencing a new genome, to identifying a suspect from a tiny drop of blood, or to diagnosing a life-altering genetic condition. Ultimately, this simple, elegant process gives us the key to the code of life itself. And once you have the key, you can open any door. Which leaves us with one last big question. Now that we can read the genetic secrets of any living thing, what secrets should we read first?